Biomolecules. Exercise. Biomolecules. Exercise pages 320 to 321. Exercise Q1.1, page 320. Select the most correct choice. CH2OHCO, CHOH, 4CH2OH is an example of 1. Aldohexose. 2. Aldoheptose. 3. Ketotetrose. 4. Ketoheptose. Solution. Ketoheptose. Exercise Q1.2, page 320. Select the most correct choice. Open chain formula of glucose does not contain 1. Formal group. 2. Anomeric hydroxyl group. 3. Primary hydroxyl group. 4. Secondary hydroxyl group. Solution. Anomeric hydroxyl group. Exercise Q1.3, page 320. Select the most correct choice. Which of the following does not apply to CH2NH2CU? 1. Neutral amino acid. 2. L. Amino acid. 3. Exists as Zwitter ion. 4. Natural amino acid. Solution. L. Amino acid. Exercise Q1.4, page 320. Select the most correct choice. Tryptophan is called essential amino acid because 1. It contains aromatic nucleus. 2. It is present in all the human proteins. 3. It cannot be synthesized by human body. 4. It is essential constituent of enzymes. Solution. It cannot be synthesized by human body. Exercise Q1.5, page 320. Select the most correct choice. A disulfide link gives rise to the following structure of a protein. 1. Primary. 2. Secondary. 3. Tertiary. 4. Quaternary. Solution. Tertiary. Exercise Q1.6, page 320. Select the most correct choice. RNA has underscore 1. A. U base pairing. 2. PSPS backbone. 3. Double helix. 4. G. C base pairing. Solution. RNA has A. U base pairing. Exercise Q2.1, page 320. Give scientific reasons. The disaccharide sucrose gives negative Tollens test while the disaccharide maltose gives a positive Tollens test. Solution. 1. The structure of sucrose contains glycosidic linkage between C1 of alpha glucose and C2 of beta fructose. 2. Since the potential aldehyde and ketone groups of both the monosaccharide units are involved in the formation of the glycosidic bond, i.e., alpha, beta-1, 2-glycosidic bond, sucrose is a non-reducing sugar and gives negative Tollens test. 3. The glycosidic bond in maltose is in between C1 of one glucose ring and C4 of the other, i.e., alpha-1, 4-glycosidic linkage. 4. The hemiacetyl group at C1 of the second ring is not involved in the glycosidic linkage. Hence, maltose is a reducing sugar and gives positive Tollens test. Exercise Q2.2, page 320. Give scientific reasons. On complete hydrolysis, DNA gives equimolar quantities of adenine and thymine. Solution. 1. Both the strands of DNA double helix are complementary to each other. 2. That is, a number of bases on each strand are equal and complementary to each other. 3. As adenine pairs with thymine, the number of adenine bases on one strand and thymine on another are equal in number. Thus, on complete hydrolysis, DNA gives equimolar quantities of adenine and thymine. Exercise Q2.3, page 320. Give scientific reasons. Alpha amino acids have high melting points compared to the corresponding amines or carboxylic acids of comparable molecular mass. Solution. 1. This is due to the peculiar structure called Zwitter ion structure of alpha amino acids. 2. Alpha amino acid molecule contains both acidic carboxyl COOH group as well as basic amino NH2 group. 3. Proton transfer from the acidic group to the basic group of amino acid forms a salt, which is a dipolar ion called Zwitter ion. Thus, alpha amino acids have high melting points compared to the corresponding amines or carboxylic acids of comparable molecular mass. Exercise Q2.4, page 320. Give scientific reasons. Hydrolysis of sucrose is called inversion. Solution. 1. Sucrose C12H22011 is dextrorotatory plus 6.5 degrees. On hydrolysis with dilute acid or an enzyme called invertase, sucrose gives equimolar mixture of D- plus glucose and D- fructose. 2. Since the Liouville rotation of fructose minus 92.4 degrees is larger than the dextrorotation of glucose plus 52.7 degrees, the hydrolysis product has net Liouville rotation. Hence, hydrolysis of sucrose is also called inversion of sucrose. Exercise Q2.5, page 320. Give scientific reasons. On boiling egg, albumin becomes opaque white. Solution. 1. Proteins when subjected to high temperature undergo disruption of noncovalent interactions which are responsible for the specific shape of protein. That is, it undergoes denaturation. 2. Denaturation disturbs the specific structure of egg albumin which causes a change in the physical properties. Thus, on boiling egg, albumin becomes opaque white. Exercise Q3.1, page 321. The following statement applies to DNA only, some to RNA only, and some to both. Label them accordingly. The polynucleotide is double-stranded. Solution. The polynucleotide is double-stranded. DNA. Exercise Q3.1, page 321. The following statement applies to DNA only, some to RNA only, and some to both. Label them accordingly. The polynucleotide contains uracil. Solution. The polynucleotide contains uracil. RNA. Exercise Q3.1, page 321. The following statement applies to DNA only, some to RNA only, and some to both. Label them accordingly. The polynucleotide contains D-ribose. Solution. The polynucleotide contains D-ribose. RNA. Exercise Q3.1, page 321. The following statement applies to DNA only, some to RNA only, and some to both. Label them accordingly. The polynucleotide contains guanine. Solution. The polynucleotide contains guanine. Both DNA and RNA. Exercise Q3.2, page 321. Write the sequence of the complementary strand for the following segment of a DNA molecule. 5 feet CGTTTAAG 3 feet. Solution. Original strand 5 feet. Dash. CGT. T. T. AAG 3 feet. Exercise Q3.3, page 321. Write the names and schematic representations of all the possible dipeptides formed from alanine, glycine and tyrosine. Solution. 1. Glycylglycine. Gly 2. Alanylalanine. Ala 3. Tyrosyltyrosine. Tyr 4. Glycylalanine. Gly 5. Alanylglycine. Ala 6. Glycyltyrosine. Gly 7. Tyrosylglycine. Tyr 8. Tyrosylalanine. Tyr 9. Alanyltyrosine. Ala Tyr. Exercise Q3.4, page 321. Give 2 evidences for presence of formal group in glucose. Solution. 1. Glucose gets oxidized to a 6-carbon monocarboxylic acid called gluconic acid on reaction with bromine water, which is a mild oxidizing agent. Thus, the carbonyl group in glucose is in the form of formyl CHO. 2. Hemiacetyl group of glucopyranose structure is a potential aldehyde, formyl group. It imparts reducing properties to glucose. Thus, glucose gives positive Tollens test or Felling test. Exercise Q 4.1, page 321. Draw a neat diagram for the following. Haworth formula of glucopyranose. Solution. Exercise Q 4.2, page 321. Draw a neat diagram for the following. Zwitter ion. Solution. Exercise Q 4.3, page 321. Draw a neat diagram for the following. Haworth formula of maltose. Solution. Exercise Q 4.4, page 321. Draw a neat diagram for the following. Secondary structure of protein. Solution. Proportional helix. Beta pleated sheet. Exercise Q 4.6, page 321. Draw a neat diagram for the following. DCMP. Solution. Exercise Q 4.7, page 321. Draw a neat diagram for the following. 1 purine base from nucleic acid. Solution. Adenine A. Guanine G. Exercise Q 4.8, page 321. Draw a neat diagram for the following. Enzyme catalysis. Solution.